Okay, so good morning once again. I will now be discussing one of the most important topics here in immunology and serology, and this will be about autoimmune diseases or autoimmunity. So we will be discussing at this point the mechanisms of autoimmunity as well as the different kinds of systemic autoimmune diseases and different kinds of organ-specific autoimmune diseases. So before that, let us define first some of the terms. So what do we mean by auto or self-antigens? So when we say auto or self-antigens, we are actually referring to antigens present in own cells. Okay, so antigens present in our own cells. So since these are antigens present in our own cells, this particular kind of antigen can be altered by the action of bacteria, viruses, chemicals, or drugs as non-self. Okay, so why is it important, the term non-self? Okay, remember, when you say self, it is our own body. And it is recognized as non-foreign. However, when we say non-self, then it is something that is recognized as foreign. Therefore, it is vulnerable to attacks. This becomes vulnerable to attacks by the cells of our immune system. Okay, so that's what we meant by non-self. So antigens present in our own cells can be altered by bacteria, viruses, chemicals, or drugs. Now, when we say autoantibodies, the autoantigen or the self-antigen will elicit now autoantibodies. Okay, and these autoantibodies will now attack the auto or self-antigens. Okay. So autoimmunity, therefore, is the immune response of autoantibody against self-antigen. So that's what we meant by autoimmunity. So humoral or cell-mediated immune response against the constituents of the body's own tissues. Okay, so there are two types of attacks that can be done to our own self. Okay, it either humoral. When we say humoral, our own antibodies will attack our own tissues. And when you say cell-mediated, okay, it could be through the action of, let's say, for example, cytotoxic T cells, natural killer cells, will be attacking our own tissues. And take note that there are actually more than 80 kinds of diseases caused by these particular autoimmune diseases. Okay, so again, just so to emphasize to you, so humoral means that our own antibodies can attack our self-antigen. Cell-mediated means that cells of the immune system, such as the cytotoxic T cells, can actually attack our own body. Okay, and it can cause tissue and organ damage. So that's the reason why there are actually two main types of autoimmune diseases. We will be discussing them specifically later on, and that would be systemic or organ specific. Now, there are different reasons why do we have autoimmune disease, and one of the reasons is the loss of self-tolerance. So when you say self-tolerance, we are referring to loss of immunologic tolerance. So what do you mean by tolerance? Ability of the immune system to accept self-antigen and not initiate an immune response against them. So we will be talking about it one by one. Okay, so these are the reasons why there are autoimmune diseases. Okay, so the first reason is sequestered antigen or hidden antigen. Okay, so when you say sequestered or hidden antigen, these are antigen in the secluded places, okay, that are not accessible to the immune system. For example, the lens antigen found in the cornea of the eye, sperm antigen, or thyroglobulin. So these are actually considered to be as sequestered or hidden antigen. And eventually, this hidden antigen can be exposed. And once these are exposed, then we will now be prone to attacks. Okay. Now, the second theory is the presence of neoantigens. Okay, what do you mean by neoantigens? Newly formed antigens. Okay, so this is what we define first. Okay, so neoantigen is the altered or modified antigens by physical, irradiation, chemical drugs, or microbial agents. So our own bodies becomes newly formed antigens whenever we are exposed to irradiation, drugs, or even microbial agents. The third reason is cessation of immunologic tolerance. Okay, so cessation of immunologic tolerance. So let us discuss this particular graph. Okay, so when we say central tolerance, okay, remember that central tolerance occurs in thymus and bone marrow as the T and B lymphocytes mature. So this is your thymus, for example. So this is your thymus, and then this is your bone marrow. If T cells, so take a look, if T cells, okay, possess TCRs. So what are TCRs? TCRs are T cell receptors. Okay, if T cells, okay, possess TCRs that binds to self-antigen in the thymus, okay, they are deleted by means of apoptosis. Okay, so if TCRs bind to self-antigen in the thymus, they are deleted by means of apoptosis. And why is it important? So that later on, they will not actually escape to the circulation and will not be treated as foreign. Alternately, they can differentiate into T-red cells. So, ito yun, no? So, some of them can be deleted by means of apoptosis. So, when we say apoptosis, we are referring to programmed self-death. Now, alternately, some of them can be differentiated into T-red cells. And what's the importance of the T-red cells? T-red cells inhibit immune responses to self-antigen. So it will inhibit immune responses to self-antigen. So that's the importance, okay, of the T-reds. Now, let's take a look for the B cells. Okay. If B cells produce BCR, again, when we say BCR, we are referring to the B cell receptors that bind strongly to self-antigen in the bone marrow. They are also deleted by apoptosis by means of negative selection. So if B cells can be deleted as well by apoptosis. So again, what's the importance of apoptosis? Apoptosis will cause programmed cell death so that these particular cells that have been exposed to self-antigen will no longer be treated as foreign by the cells of our immune system. Okay. Alternatively, they can edit their receptor, so they are no longer antigen-specific, or they would downregulate the expression of their receptor. So they become unresponsive to an antigen. So this is what you call anergy. Okay. What's the first step to anergy? Receptor editing. So if there's a receptor editing, it means that there is a downregulation. So they will now possess the characteristic called anergy. So when you say anergy, it's a very important way to prevent autoimmune disease because they become unresponsive to antigen. Okay. Now, there is what you call self-reactive T cells and B cells that have escaped the secondary lymphoid organs like lymph node and spleen. Ito yun. There are some T cells and B cells that have escaped, okay, the secondary lymph nodes such as the lymph node and spleen. And they are actually being removed by means of what? Peripheral tolerance. Okay? So, peripheral tolerance. So, cells that are being removed to become non-reactive to self-antigen, if they are being removed in the bone marrow and thymus, we call it central tolerance. If they are being removed in spleen, lymph node, and other secondary lymphoid organs, we call it peripheral tolerance. Okay, so take note, what's the importance of peripheral tolerance? Peripheral tolerance of T cells. Okay, these are the peripheral tolerance, no? Peripheral tolerance of T cells can result from inhibition by Tregs, lack of co-stimulatory signal from antigen-presenting cells, binding of inhibitor receptors like CTLA4, or death by apoptosis. So again, pwedeng magkaroon ng apoptosis pa din. Okay, self-reactive B cells in the periphery can also be deleted by means of apoptosis. Okay, so, or pwede silang maging anergic. Again, pag sinabi nating anergic, anong ibig sabihin nun? Non-responsive. So, dalawang, ibiruin mo, our immune system is very capable. No? Our immune system is very capable. Nakalusot sa central tolerance, meron tayong peripheral tolerance. Nakalusot sa peripheral tolerance, then, what will happen? If our cells escape the peripheral tolerance, this is where autoimmune disease will take place. Okay? So, why do you think autoimmune disease takes place? Because the central and peripheral tolerance fail. So, for some individuals, the peripheral and central tolerance fail. This is where we get autoimmune disease. Okay? So that is the third cause of autoimmune disease. Number 4 is the presence of cross-reacting antigen. Okay. A foreign antigen which resembles self as second antigen. So those antigens that would mimic, okay, those antigens that would mimic our tissues. So they become cross-reacting antigens. So many species, many bacteria share organ-specific antigens. So for example, do you know that the antigen of human brain and the antigen of sheep brain are almost have the same characteristic? Do you know that the M protein found in Streptococcus would have the same characteristic as our heart muscle? Meaning to say, if we are infected with Streptococcus, there is also a big chance for us to get endocarditis. And the cells of our immune system will even destroy our heart cells, our heart muscle. Okay, do you know that if you are infected with Streptococcus, okay, the nephrogenic strain of Streptococcus, the one that can cause acute glomerulonephritis, share similar epitopes with the renal glomeruli, with the glomerulus of our kidneys. Meaning, if you are infected with nephrogenic strains of Streptococcus, there's also a chance that the cells of your immune system will even be attacking your own kidneys. So that's a possibility. And of course, the fifth one, which I've already discussed, is the loss of self-tolerance. Okay, this is when there is an overactivity or lower activity of a T and B cells. So meaning to say you lost both the central and peripheral tolerance. Okay, now this particular slide here will explain to us, okay, the genetic and environmental factor, okay, as a result of autoimmunity. So it says here that autoimmunity is thought to result from complex interaction between the genetic and environmental factors. So ano ba yung mga environmental factors? Okay. Female hormones, tissue injury and release of self-antigens, microbial antigens that are capable of molecular mimicry, epitope spreading, superantigens, stimulation of immune responses, and epigenetic factors, which includes toxins, food, drugs, aging, and post-translational modifications. Now, the thing is, inheritance of certain genes makes some individual more susceptible to a particular autoimmune disease as compared to other individuals who do not have that particular kind of genes. Okay, so this particular characteristic is what we call genetic predisposition. Okay, so we call it genetic predisposition. Okay, so genetic predisposition means that specific HLA type can determine the effectiveness of antigen presentation and non-MHC genes which influence the development and regulation of immune response. So, combining this factor, so this is factor 1, plus you are genetically predisposed, okay, plus hormones. For example, you have hormones. Do you know that women are more likely are actually more prone to have autoimmune disease? You know why? Because hormones can also influence autoimmunity. Sabi nga rito, women are almost 3 times as likely to acquire an autoimmune disease as compared to men. Okay. Tapos, nagkaroon pa ng tissue injury. Tissue injury can, for example, can expose hidden antigens in the lens, can expose sequestered antigens. So, tissue injury can lead to release of cryptic antigens. Pag sinabi natin cryptic antigens, these are hidden antigens, which can induce an autoimmune disease. So, these self-antigens were hidden in the tissues of the host during the early development of T and B cells. So the tolerance was not developed against them. Kaso nga lang, na-exposed sila because of the injury. Tapos ito pa, yung microbial infections, which may also trigger autoimmunity in several ways. So one way is through molecular mimicry. Ito yung in-explain ko sa inyo kanina, whereby bacteria or viruses possess antigens that are very similar to our self-antigen. So a second way is through epitope spreading. So when you say epitope spreading, this is where the immune response to a microbe, immune response to a microbe expands to activate immune response to other antigens, including self-antigen. And the third way is a super antigen. Super antigen, the reason why we call it super antigen, it's primarily because it can activate numerous clones of T cells through certain viruses, which can cause polyclonal B cell activation. And the third factor, the last factor is epigenetic modification. This is when we are exposed to environmental factors that can alter gene expression, which may also contribute to autoimmunity. So imagine having all of these factors, tapos babae ka pa, tapos you are genetically predisposed, then nagkaroon pa ng loss of immunologic tolerance, hence nagkaroon na siya ng autoimmune disease. Okay, so that's the advantage, no? That's the not really the advantage, but that's the factor for the development of autoimmune disease. But does it mean yung mga guys, males, hindi na sila magkakaroon ng autoimmune disease? Actually, pwede pa rin. I'm telling you that females are just more vulnerable, but males can still develop autoimmune disease. Okay. So there are more than 100 autoimmune diseases discovered. Okay? And they are divided into two, systemic, and the other one is organ-specific. So we will be discussing these kinds of autoimmune diseases. Actually, there is a third classification. Okay, so yung localized, this is what we call organ-specific. And then we have the systemic, and then we have the hemolytic autoimmune disease. So yung hemolytic autoimmune disease is actually a clinical disorder due to the destruction of blood components. Usually, when we develop autoantibodies against one's own RBC. So for example, there are some people who develop an autoimmune thrombocytopenia. So kapag meron kayong autoimmune thrombocytopenia, your platelets becomes very low as a result of antibodies against your own platelets. And these antibodies were produced by your own body. So that's what you call the hemolytic autoimmune disease. So when you say localized autoimmune disease, this is also known as the organ-specific autoimmune disease, meaning to say only a particular organ is affected due to autoantibodies. So for example, thyroiditis attacks the thyroid. If you have multiple sclerosis, it's attacked the myelin sheath, coating of nerve axons. If you have myasthenia gravis, it attacks the nerve-muscle junction. If you have the juvenile diabetes or the type 1 diabetes mellitus, it attacks the insulin-producing cells of the pancreas, particularly the islet of the Langerhans of the B cells of the pancreas. Okay, so these are the different examples of organ-specific autoimmune disease. And then the third one is the systemic, meaning to say it is no longer organ-specific because immune complex, okay, take note, we discussed already about immune complex or the type 3 hypersensitivity reaction, remember that? So immune complex accumulate in many tissues and this one causes inflammation and damage. So this particular type of autoimmune disease may affect many organs or the entire body as well. So what are the examples of systemic autoimmune diseases? Examples include SLE, the systemic Lupus Erythematosus. So later on, we will be discussing about anti-nuclear antibody and why do we call it systemic? Because it affects our hands, I'm sorry for the typographical error, kidneys, heart, brain, lungs, and skin. Okay, it harms the kidneys, heart, it's not a typo, it harms the kidneys, heart, brain, lungs, and skin. Rheumatoid arthritis, if you develop an anti-IgG antibodies, okay, or the rheumatoid factor, it affects the joints, heart, lungs, nervous system. Rheumatic fever is a cross-reaction between antibodies to Streptococcus and autoantibodies. So these are all examples of systemic autoimmune disease or non-organ specific autoimmune diseases. Okay, so what are the criteria, okay, to be considered as autoimmune? Okay, first is autoimmune response is regularly associated with the disease. And we can induce a replica of disease in the animal. It is immunopathologic changes can be observed in the natural and experimental diseases, and they should parallel each other. Meaning to say, if you replicate it in the animal, you will be able to observe the same immunopathologic changes. The disease can be transferred via exchange of serum or lymphoid cells. Okay, so autoimmune disease can be transferred. So this is what I told you about the genetic predisposition. So if you have HLA-DR4, you are at risk of getting, you are genetically predisposed to have rheumatoid arthritis. If you have HLA-DR5, you are genetically predisposed to have thyroiditis. If you have HLA-DR2, you are genetically predisposed to have multiple sclerosis. If you have HLA-DR3, you are genetically predisposed to have systemic lupus erythematosus. And alongside with HLA-P8, then you are genetically predisposed to have Graves' disease. So these are just some of the few examples of genetic predisposition of people to have autoimmune diseases. So what should be the general signs? First sign is that there would be elevated levels of gamma globulin. So antibodies will be increased, and there will be various types of autoantibodies. Number 3 is depressed level of serum complement. Why? Because serum complement is already deposited, okay, as part of immune complex. And that is the reason why serum complement is actually decreased. Okay, number 4, there is an immune complex in the serum. Number 5 is there is a depressed level of suppressor T cells, or this is what you call the Treg. And there would be lesions detected in the biopsy. Okay, so these are the general signs of having autoimmune diseases. Okay, so let's talk about them one by one. So the first type of autoimmune disease that we will be discussing is known as the SLE, or the systemic lupus erythematosus. So this kind of autoimmune disease is a chronic systemic inflammatory disease that affects multiple organ systems. So here, we develop autoantibodies, mostly known as anti-nuclear antibodies, because these antibodies are directed against, for example, double-stranded DNA and other components, lymphocytes, RBCs, and platelets. So immune complex is formed. By the way, when we say immune complex, okay, so these are made up of antigen, antibody, and complement. Okay, so that's what we meant by immune complex. So triggering complement activation, chemotaxis of neutrophils, and inflammation. Okay, so that is systemic lupus erythematosus. So what are the clinical symptoms of SLE? So there would be joint involvement, skin rashes, okay, renal involvement, so kidneys can be destroyed as well, neurologic symptoms, anemia, leukopenia, thrombocytopenia, and others. So ibig sabihin, all of the blood cells are decreased. So if the blood cells are decreased, we call it pancytopenia. Okay, so these are the signs of SLE. So you could see that it's really systemic, because pleural effusions in the lungs, there would be heart problems, kidneys will be affected, arthritis, and then you'd be able to see the butterfly rash. This is the rash in the skin, and the joints can be affected as well. So we call it the Raynaud's phenomenon, okay, for SLE. Okay, so in CNS, there's a possibility of seizures in the skin. There would be a butterfly rash in the heart. There could be endocarditis and myocarditis in the kidneys. There could be glomerulonephritis. And then in the lungs could have pericarditis, pleuritis. There could be hematologic effects, hemolytic anemia, leukopenia, thrombocytopenia, arthritis, or the Raynaud's phenomenon, and even lymphadenopathy. So see, SLE is considered to be a systemic, because it's a multi-organ involvement. Okay, so this is another example of butterfly rash. So another example of butterfly rash in SLE. Okay, and here you would see that immune complex is deposited. Okay, let's say for example, this is a major blood vessel, so it's actually it's actually very crucial. So immune complex is made up of antigen, antibodies, and then it will be attacked by the complement. So one way of diagnosing SLE is to get the Buffy coat of the patient. So if you could remember, Buffy coat is actually made up of PAC-RBC, and then this is the WBC, the creamy white layer, and then this is the plasma. So the creamy white layer in between the PAC-RBC and the RBC, and then I mean the creamy white layer between the PAC-RBC and the plasma, that's what you call the Buffy coat. This is where we can find the LE cells. Okay, so LE cells are actually a special type of WBCs where in the nucleus, the one that you can see here, are actually being pushed away at the periphery or at the side. So that's the characteristic of the LE cells. So that's one way of diagnosing SLE. So aside from CBC urinalysis, there would be elevated CRP and ESR. Again, CRP and ESR are non-specific indicator for inflammation. We can also quantify complement because C3 can be decreased during inflammation. And most importantly, the determination of anti-nuclear antibodies, ANA. So determination of anti-nuclear antibodies can be used for the diagnosis of SLE. So what are anti-nuclear antibodies? When you say anti-nuclear antibodies, these are directed against antigen found in the cell nuclei. So if this is the cell, and then this is the nucleus, so antibodies will be attacking the antigen found here. So that's what we meant by anti-nuclear antibodies. And do you know that 95% of lupus patients would have anti-nuclear antibodies? However, anti-nuclear antibodies is common to SLE, but it is not specific to SLE. So this is what you need to remember, okay? It is common to SLE, but it is not specific to SLE. So anti-nuclear antibodies would include anti-double-stranded DNA. This one is the most specific to lupus. Anti-single-stranded DNA, anti-histone and nucleosomes, antibodies to centromere or nucleolar components, and anti-ENA. Okay, so ANA therefore are also known as anti-nuclear factors. So these are autoantibodies that bind to the content of the cell nucleus. So most especially in normal individuals, so even if you're normal individuals, the immune system produces antibodies to foreign proteins, but not to human proteins. If you are in normal individuals, you should only be reacting to foreign proteins, but never to your own proteins. Okay. In some individuals, antibodies are produced to human antigens. So this is where anti-nuclear antibody will come into play. So there are many subtypes, which I have already enumerated a while ago. So proteins, nuclear proteins. So if you develop antibodies against nuclear proteins, this is where you develop anti-nuclear antibodies. Proteins, nuclear proteins, by the way, are proteins that have been synthesized in the nucleus and therefore were distributed to their respective sites in the cell. So let's say, for example, if this is the nucleus, if this is where you can find double-stranded DNA. So this is where you can find anti-RO, Na, SNP, RNP, JO1, STL7, and nucleosomes. So you develop antibodies against different kinds of nuclear proteins. So, how do we detect anti-nuclear antibodies? Okay, if you remember our labeled immunoassay, it is detected by means of indirect immunofluorescent assay, or IFA. It is a method for screening and titration of circulating autoantibodies in human serum. And we are using 3 different tissues from rats, namely from the liver, kidneys, and stomach. So it enables autoantibodies to be more easily identifying by comparing the results obtained with each tissues. So there are 3 major antibodies detected, okay? We have the anti-nuclear antibodies, anti-mitochondrial antibodies, and anti-smooth muscle antibodies. So an indirect immunofluorescence technique is utilized. And what is our sample? Our sample is patient serum. And what are we detecting in the patient serum? We are detecting anti-nuclear antibodies. So serum plus controls are incubated with the substrate in the slides. So if you do not have anti-nuclear antibodies, you call that unreacted antibodies, and eventually this particular unreacted antibodies will be washed away, and appropriate fluorescence label conjugate is applied. Unbound conjugate, meaning to say those who are not able to form an immune complex, will be washed off, and the slides are viewed with fluorescence microscope. So a positive sample will produce an apple green fluorescence. So this is the principle, no? So this is now your serum. So your serum is here. So if you are positive with anti-nuclear antibodies, this is now the antibodies from patient. And then you will be adding anti-human globulin that is fluorescent labeled. Why do we need it to be fluorescent labeled? We need it to be fluorescent labeled so that it will glow in the dark when observed under the fluorescent microscope. And these are the interpretation. So you will now interpret the cells. This is how homogeneous look like. This is rim. You can actually see that fluorescence are seen at the site. So you call that rim. Speckled. And then you have the nucleolar, and then you have the centromere. Okay, so these are the 5 interpretations, okay, in anti-nuclear antibodies. So you have homogeneous, rim, speckled, nucleolar, and centromere. So nucleolar, rim, speckled, homogeneous, okay, so, and then we have the centromere. Why is it important? Because by looking at the pattern, you can determine what antigen is present and what diseases could be associated with it. So for example, if you are able to observe homogeneous, meaning to say, when you say homogeneous, all throughout the cells, the pattern of fluorescence is the same. The pattern of fluorescence is the same all throughout the cell. Okay, so you call it homogeneous. So it means that you are able to see double-stranded DNA, and the disease that is associated with it could be SLE, mixed connective tissue disease, drug-induced lupus. Okay, peripheral. You remember the peripheral? The fluorescence are actually more concentrated at the side. So that is also known as rim. Okay, so common antigen involved would be native double-stranded DNA. And having a rim or peripheral pattern is the most specific for SLE. Speckled. What's the difference between speckled and nucleolar? Okay, speckled are actually smaller dots as compared to nucleolar. That's speckled. These are smaller. as compared to nucleolar. Okay, so speckled means that you're able to see or extractable nuclear antigen, nuclear protein, SCL70 and SSP. So this one is actually more specific for scleroderma, Sjogren's syndrome, mixed connective tissue cell disease, and SMA. And then the nucleolar have a bigger dots. They have bigger dots as compared to speckled. So this is an indicator for the presence of 4 to 6S sRNA. Okay, so it's the diseases that are associated are scleroderma, SLE, rheumatoid arthritis, Sjogren's syndrome, progressive systemic sclerosis. Okay, so what's the importance of anti-nuclear antibodies? It is a serologic test of patients' systemic autoimmune disease. Okay, it can provide further diagnostic and prognostic data concerning patients who have minimal symptoms or who have clinical features of more than one autoimmune disease. As you can see a while ago, where it is not specific, except if you are able to see the Rheum, because Rheum or peripheral is the one that is most specific for systemic lupus erythematosus. Now, immunofluorescence using a parasite, not really a parasite, but an organism known as Cryptidia luciliae. So it is actually an example of a trypomastigote. Okay, we can actually use this as a substrate. Okay, so instead of using the rat kidney cells as substrate, we can use a particular organism. So it contains a singular organelle called kinetoplast. So kung naalala niyo yung kinetoplast, this is where the flagella will actually come from. Okay, it is used to detect antibodies to double-stranded DNA. So this one is actually more specific for SLE as compared to other animal substrate, because before, the one that I've shown you before, those substrate, those cells came from rats. But this one, it came from a protozoan. Okay, protozoan known as the trypomastigote. Okay, now aside from that, we can also use the Auster-Loney technique. Remember the Auster-Loney technique? Okay, the Auster-Loney technique can detect a specific ENA. Okay, so extranuclear antibodies. Okay, so immunodiffusion test detects antibodies to the specific ENA. So just to remember what is the principle of Auster-Loney test. The principle of Auster-Loney test is double diffusion. And there are actually three interpretations that you can determine. There are actually three interpretations that you can determine in this particular type of test. By the way, let me correct myself. It's not extracellular. ENA stands for extractable nuclear antigen. So I stand corrected, okay? So there are three possible interpretations. So if you can remember, in Auster-Loney, you can have identity, partial identity, and non-identity. So for example, in this particular diagram, let me enlarge this photograph. Patient A, so this is patient A, this is patient A, and then this is patient B. Okay, so at the upper portion, you have added anti-SM antibody. At the lower portion, you have added anti-RNA antibody. So patient A has anti-SM antibody. Why did I say that? Because of this particular arc formation. Okay? And that particular one is identified with the anti-SM positive control, because this is the control. Anti-RNP is also, this is the control for anti-RNP. Okay. There is partial identity with anti-RNP positive antibody because the center well contains free, as you can see here. Let me enlarge again this particular photo. You can see the central well, it contains free anti-SM, okay, as well as SM-RNP complexes. Okay, so therefore we can see that both reacts with anti-SM, but it's not totally the semi-arc formation. This particular formation is known as the spore formation. And since it is a spore formation, it is called anti-RNP antibody. So in this example, the patient has antibody to SM, but partial identity with anti-RNP antibodies. So ENA, therefore, can be useful this one can be useful with extractable antigen. So again, let me interpret it again. Patient A has anti-SM antibodies. So this is the anti-SM control, and there is a cross formation. There is a partial identity with the anti-RNP positive control. So this is the anti-RNP positive control, and you can see the spore formation because the central well contains free SM antibodies. Where is the central well? This is the central well. Okay, both react with anti-SM in the patient's serum. So yung ENA kasi has, etong central well kasi has both anti-SM and anti-RNP. But the only one that has a arc formation is anti-SM antibody. And here, it doesn't have arc formation. Therefore, in this patient, in this particular example, patient A has antibody to SM. Okay, so that's the interpretation of extractable nuclear antigen Austerlonites. So what's the limitations of RNA? Okay, their presence does not mandate the presence of illness since they can be found in otherwise normal individuals. See what I told you. ANA is not specific for SLE, but it is commonly found or commonly increased in SLE. Accurate interpretation of different nuclear patterns is confounded by several difficulties, such as one nuclear pattern may be obscure and prevent the recognition of another pattern if antibodies are present simultaneously. So in one particular test, you'd be able to see, let's see, for example, you'd be able to see nucleolar, you'd be able to see homogeneous, you'd be able to see ring in one particular slides. Okay, so that's the disadvantage. Okay, so aside from that, we can also have the so-called phospholipid antibodies. Okay, phospholipid antibodies can be found in approximately 60% of lupus patients. Okay, meaning to say it is associated with deep vein and arterial thrombosis. So phospholipid antibodies will give you an increased risk of recurrent pregnancy loss. Ito yung mga sa pregnant women, they call it the APAS. If a pregnant woman is positive for APAS, if a pregnant woman is positive for APAS, so most likely she'll not be able to complete the 9 full term. Okay, so the clinical significance of APAS is that it can cause false positive result in non-treponemal tests for syphilis. And since it is considered to be as a lupus anticoagulant, it will produce prolonged APTT and PT, activated partial thromboplastin time and prothrombin time. So these are examples of coagulation studies. So what's the significance of APAS? Again, it is found in majority of lupus patients. It will make you at risk of having deep vein or arterial thrombosis. And if you are a woman, it will make you vulnerable to have miscarriage. Okay, and it will also cause false positive in non-treponemal tests for syphilis, such as the VDRL and RPR. Okay, so let's talk about this time a different kind of autoimmune disease. So we're done with SLE. Now let's talk about rheumatoid arthritis. So rheumatoid arthritis is actually caused by chronic arthritis of the peripheral joints that can progress to joint deformity and disability. Unfortunately, 25% of patients have actually developed osteoporosis. And some patients would even develop subcutaneous nodules, pericarditis, interstitial lung disease, and even vasculitis. So vasculitis is the inflammation of the blood vessels. This one is the inflammation of the pericardium of the heart muscles. So again, the joints are actually damaged. So this is an example of a synovial joints. Okay, so we're in the particular connective tissue or cartilage here is actually being eroded. Cartilage is very important so that the bone will not actually friction against one another. And that particular, these are the ones that are being damaged in cases of rheumatoid arthritis. Okay, so there is a pannus formation. Okay, so that's one of the characteristics. And the boutonniere deformity of the thumb. So you'll be able to see the ulnar deviation of the metacarpophalangeal joints and the swan neck deformity of fingers. So may nakita kayong mga joints. Okay, so these are characteristics of the late stage of rheumatoid arthritis. And some of the patients would even develop osteoporosis. Okay? So again, there is a destruction of cartilage. That's why it's very painful. So joint pain occurs at various joints. So the pathology and treatment. So there is an inflammation, and inflammation destroys the bone and cartilage. The alpha tumor necrosis factor plays a key role in the process. So the alpha necrosis tumor factor, the reason why they play a key role is that they can actually overly active osteoclast will absorb the bone. So autoantibodies combine with antigen to form the immune complex. Okay, so immune complex form between the autoantibodies produced in rheumatoid arthritis. So one of the antibodies that you can actually hear is the anti-CCP. So this increase inflammation by causing type 3 hypersensitivity reaction. So ibig sabihin, ng type 3 hypersensitivity reaction, there is already an immune complex. Okay. So, ano yung mga different diseases? Non-steroidal anti-inflammatory drugs or NSAIDs are actually just being used to avert inflammation, but it will not cure rheumatoid arthritis. And then there is also called drugs such as DMARDs. So ano ba yung example ng DMARDs? So disease-modifying anti-rheumatic drugs. So this includes methotrexate. Methotrexate will reduce inflammation and it will prevent disease progression. Now, a while ago, I told you that alpha tumor necrosis factors will play a key role in the process and development of rheumatoid arthritis. Okay? Alpha tumor necrosis factors, there are biologic agents kasi that can actually target on alpha tumor necrosis factor. First biologic agent would be the presence of monoclonal antibodies. So monoclonal antibodies can destroy our alpha tumor necrosis factors. And the second one is the receptors for alpha tumor necrosis factor. So these particular receptors will fuse with an IgG molecule, which binds to stimulating alpha tumor necrosis factor. Okay. So in the process, rheumatoid factor can be found in patients. So what is rheumatoid factor? So if you are positive for rheumatoid factor, if you are positive with rheumatoid factor, it means that you are more likely you have an autoimmune disease. So rheumatoid factor is an autoantibody. And most of the time, this autoantibody is IgM that reacts with the Fc portion of the IgG. So for example, this is our IgG. And then IgM will attack the Fc portion of our IgG. And it is found approximately in 80% of patients with rheumatoid arthritis. Okay, so it is found approximately in 80% of patients with rheumatoid arthritis. So remember, guys, rheumatoid factors is not specific, okay, for rheumatoid arthritis. Only 80% of patients with rheumatoid arthritis would have rheumatoid factor because it can also be found in other connective tissue diseases such as SLE, Sjogren's syndromes, scleroderma, and mixed connective tissue diseases, as well as people with some chronic infections. So rheumatoid factor assays includes latex agglutination, nephelometric methods, ELISA, and chemiluminescence assay. Okay, now there is one particular antibodies that are also highly specific for rheumatoid arthritis. We call it the anti-CCP. So CCP stands for antibodies against cyclic citrullinated peptide. So the peptide here contains a modified arginine. So anti-CCP assays are performed mainly by ELISA. So there are many in our laboratory. We have rheumatoid factor, no? That can be, the most common is latex agglutination for rheumatoid factors, but there are other assays. There are other assays, no? Pwedeng ang nephelometric for rheumatoid factor. It could be ELISA. It could be chemiluminescence assay. Okay, but recently they found out that anti-CCP is more specific. Antibodies against citrulline or cyclic citrullinated peptide. So what is a citrulline? So antibodies against a citrulline. Citrulline is an atypical amino acid. And it is usually generated, so this particular atypical amino acid is generated with peptidyl, arginine, deaminase. And this particular peptidyl arginine deaminase will what? Will modify arginine. Okay, and it modifies arginine by replacing the NH2 group. So usually, amino acid arginine should have NH2, but NH2 is replaced with with a neutral oxygen. Okay, so that's the reason why it is now a modified amino acid. So another way of testing for rheumatoid arthritis would be by means of anti-nuclear antibodies. So anti-nuclear antibodies, like what I told you, this one is not really specific for SLE. So anti-nuclear antibodies are found approximately in 40% of patients with rheumatoid arthritis. Okay? And they're commonly directed against RNP. So pag meron kayong nakita na anti-RNP, so most likely rheumatoid arthritis yan, and they will produce a speckled pattern. Remember how a speckled pattern would look like? So ESR, CRP, and complement can also be used as a general indicators. So again, ESR and CRP are not specific, okay? So these are not specific, but they are considered to be as general indicators for rheumatoid arthritis. Okay, so that ends our rheumatoid arthritis. So let's discuss another example of autoimmune disease. This time, let's talk about granulomatosis with polyangitis, otherwise known as the Wegener's granulomatosis. It's a very rare autoimmune disease involving the inflammation of, this one involves the inflammation of small to medium-sized blood vessels and respiratory tract. So it starts with blood vessels. However, it will progress to a more systemic diseases since it will also involve other organs such as the kidneys, joints, and skin. So most patients have antibodies to neutrophil cytoplasmic antigen such as proteinase 3. So here, you will develop anti-proteinase 3. So what will happen? Neutrophil will be activated and it will result in damage to vascular endothelium as a response to T helper 1. So to diagnose this particular kind of autoimmune disease, then we have to look for the presence of ANCA, ANCA. So ANCA refers to anti-neutrophil cytoplasmic antibodies. So ANCA, or the anti-neutrophil cytoplasmic antibodies, these are produced against proteins in neutrophil granules. So it is strongly associated with syndromes involving vascular inflammation. So how do we detect ANCA? We detect ANCA by using indirect immunofluorescence assay. So usually on ethanol fixed leukocytes, ELISA, or CLIA, or chemiluminescence assay. So in ANCA, sorry, I wasn't able to move my slides a while ago. In ANCA, it is detected by indirect immunofluorescence assay. So in ANCA, we are using patient serum. So we are using patient serum, and we are also using fluorescent isothiocyanate conjugate labeled anti-IgG. Okay, and then we add it together, and then we look for the presence of neutrophils that glow in the dark. So these are examples of neutrophils showing p-ANCA staining. So you can see lymphocytes here do not glow in the dark. Okay, but the neutrophil here shows a pattern. Okay, so this is actually what you call the p-ANCA fluorescence. Okay, so what's so special about the p-ANCA? Okay, so we call it the p-ANCA pattern. So the p-ANCA pattern is actually fluorescence surrounds the nuclear lobes. So you have to look for the presence of fluorescence in the nuclear lobes, blending them together. So the antibodies responsible for this pattern are positively charged antigens. So antibodies against positively charged antigens, okay, such as the Myelo peroxidase that migrate out of the granules toward negatively charged nucleus. Okay, so ANCA is the same as RIM in ANA. So same as RIM or peripheral pattern. Okay, so this time let's talk about the thyroid, autoimmune thyroid diseases. So normally, the production of the hormones T3 and T4 by the thyroid gland is carefully regulated by a feedback loop called thyroid axis. So this is the negatively feedback loop called thyroid axis. So two possible things can happen. So you can either have hypothyroidism and the other one is hyperthyroidism. So the hypothalamus secretes, so where is the hypothalamus? It's actually in the brain. The hypothalamus secretes TRH. So this is the TRH. Okay, the hypothalamus secretes TRH, and TRH stands for thyrotropin releasing hormone. What's the importance of TRH? TRH stimulates the pituitary gland to induce the release of TSH. Okay, so what is TSH? TSH stands for the thyroid stimulating hormone. Okay, so what's the function of the TSH? The TSH, on the other hand, will stimulate the thyroid gland to produce T3 and T4. So from the thyroglobulin, from the breakdown of thyroglobulin, we will now be producing T3 and T4. Okay, through the enzyme TPO. What is TPO? TPO stands for thyroid peroxidase. So thyroid peroxidase will oxidize the iodine ions. So iodine, this is the iodine ion, okay? The iodine, it will oxidize the iodine ion that are incorporated into thyroglobulin. Kaya yung mga bata na mayroong iodine deficiency would also have abnormal thyroid hormone. Now, kaya nga sabi, mag-iodize so, di ba? Now, if the levels of T3 and T4 becomes too high, so of course, as part of our regulation of the feedback loop, they inhibit the release of TRH and TSH to decrease further the thyroid hormone production. However, in cases of Graves' disease, autoantibodies to TSH, okay, increases. Okay, so autoantibodies to TSH thyroid hormone synthesized bypassing the thyroid axis. So, ay nangyari in cases of the Graves' disease. In cases of Hashimoto Thyroiditis, autoantibodies to TG and TPO lead to a decrease in thyroid hormone synthesis. So, cytotoxic T cells can also damage the thyroid gland. Okay, so in Graves' disease, therefore, there is a what? In Graves' disease, it's stimulatory. So there would be hyperthyroidism. In Hashimoto thyroiditis, okay, there is a decrease in thyroid hormone synthesis. Therefore, there is a hypothyroidism. Okay, so let's talk about first Hashimoto Thyroiditis. Hashimoto Thyroiditis is the destruction of the thyroid gland which produces hypothyroidism. So Hashimoto's is the most common autoimmune disease, and it is mostly seen in middle-aged women. So this is what you call goiter. Okay, goiter is an enlarged thyroid gland in this disease, and it is irregular and rubbery. So symptoms include, diba, fatigue, dry skin, weight gain, brittle hair, formation of a goiter. So immune destructions caused by the cytotoxic T lymphocytes will damage the thyroid cells, and production of autoantibodies that induce inflammation by binding with complement. So here, you would expect that the free T4 becomes very low kasi nga it is unbound to protein. Anti-TPO is the best indicator. Okay, so remember, this one is the best indicator for Hashimoto's thyroiditis. Then anti-TG can also be used, but it is less sensitive. However, even if they are less sensitive, it is very specific. Very specific. Okay, they are found in fewer Hashimoto's patients and in healthy persons as well. So again, characterized by enlarged thyroid gland, okay, hypothyroidism, lesions with experimental animals are after administration of Freud adjuvants are also seen in Hashimoto's thyroiditis. Okay, so let's talk about the Graves' disease. Okay, Graves' disease is characterized by hyperthyroidism. So it is an autoimmune thyroid disease, but this time it is characterized by hyperthyroidism. Do you know that it is present most often in women, especially in their 50s and 60s? So the goiter in Hashimoto thyroiditis is jelly-like, but this time the goiter in Graves' disease is firm, and it is characterized by exophthalmos. This is the exostamia. Exostamia is the hypertrophy of the eye muscle that causes the eyeball to bulge out. So lumuluwa ang mata. So orbital fibroblasts. These are the connective tissues found in the eyes. Okay, will express TSH receptors that can bind to TSH. Okay, so usually you produce antibodies against the thyroid receptor. So we call it the TRAPs. Okay, autoantibodies to thyroid stimulating hormone receptor. So do not forget this one, TRAPs represents autoantibodies to thyroid stimulating hormone receptors. So the TRAPs will actually bind to TSH or the thyroid stimulating hormone receptors. Okay, it will mimic the action of TSH on the thyroid gland, but since this bypass the thyroid axis, so ano nangyari? There would be excessive production of T3 and T4. So the presence of TRAPs is highly indicative of Graves' disease, and this can be detected by immunoassays or bioassays. So aside from TRAPs, we can also determine the presence of low TSH, high FT4, and high antibodies to TPO and TG. Okay, so Aside, it is characterized by fatigue, nervousness, increased sweating, weight loss, heat intolerance, increase in B cells, decrease in suppressor T cells. So, you are also genetically predisposed if you have HLA-B8 and HLA-BR3. I think I have already explained this already to you. So, Graves' disease is a common cause of hyperthyroidism, as what is shown here. An overproduction of thyroid hormone, which causes enlargement of the thyroid and other symptoms such as exophthalmos, heat intolerance, and anxiety. So again, this is the exophthalmia in Graves' disease. Okay, so let's talk about type 1 diabetes mellitus. Okay, so the type 1 diabetes mellitus is another example of autoimmune disease. It is an endocrine disorder characterized by hyperglycemia. So again, when you say hyperglycemia, it is an increase in high level or an increase in blood sugar level. So type 1 is characterized by the destruction of beta cells in the islet of the Langerhans in the pancreas, which will result in deficiency. Now, type 1 is actually different from type 2. Kasi in type 2, patient develop resistance in insulin due to obesity or other factors. But type 1 was previously known as the juvenile onset diabetes because it usually develops in children or even in young adults at the age of 30. So what is the importance of insulin? So insulin works by promoting the absorption of glucose by skeletal muscle or fat tissue so that it can be converted into energy. Okay, so why do we consider type 1 diabetes mellitus as an autoimmune disease? Because of the destruction of the beta cells. Okay, destruction of beta cells occur via Th1 response and it will release cytokines and inflammation. So autoantibodies can affect the regulation of pathways needed for insulin secretion. So what is the treatment? Meron palang treatment, no? Daily insulin injection to control the levels of blood glucose. So experimental treatments includes immunosuppressive drugs, biologic agents, transplantation of pancreatic beta islet cells or stem cells. Okay, so laboratory diagnosis of type 1 diabetes mellitus can also be determined using the HbA1c. Okay, so HbA1c stands for glycosylated hemoglobin. So why is it important? Glycosylated hemoglobin is actually used to monitor long-term glucose concentration for about 2 to 3 months. Okay. Now, as what I've told you, that type 1 diabetes mellitus considered as autoimmune disease because you would develop autoantibodies to GAD, which stands for glutamic acid decarboxylase 65 antigen. So that's the reason why it is considered to be as an autoimmune disease. And sometimes patients would also develop antibodies to IA2. IA2 means insulinoma antigen 2. Okay? So what is an IA2? insulinoma antigen 2 is actually considered to be as the islet cell antibodies. Now remember, diabetes mellitus, insulin is being produced at the islet of Langerhans, which is found at the beta cells of the pancreas. So islet cells antibodies are known as the ICA. I'm sorry. Islet cell antibodies is also known as the ICA. IA2 is the insulinoma antigen. So these antibodies, all of these antibodies can be tested to confirm presence of type 1 diabetes mellitus. And of course, you have to correlate it with elevated blood glucose plus the results of the HbA1c or the glycosylated hemoglobin. Okay, so this time, let's talk about the celiac disease. So it's another type of autoimmune disease, the celiac disease. So what is a celiac disease? Celiac disease affects the small intestine. It affects the small intestine and other organs. Okay, so particularly whenever you eat something with gluten. Okay, so if you will eat something with gluten, so gluten is actually a complex. Usually, if you eat food that is made up of wheat, barley, and rye, you'll have gluten. Why? Because in the gluten, there's a particular component of gluten called gliadin. So in gluten, you have a gliadin. So gliadin is the component of gluten that is resistant to digestive enzymes. So what does it mean? It cannot be broken down. Okay, so these digestive enzymes are supposed to be found in the stomach, the pancreas, and the small intestine. So once this gliadin is undigested, okay, it can trigger an autoimmune disease or autoimmune response if it passes through the epithelial barrier of the intestine. Meaning to say, if gliadin goes out of the intestine, then it will trigger autoimmune response. Okay? So one of the manifestations that you have celiac disease is the presence of TTG IgA. Okay, antibodies against TTG. TTG is an intestinal enzyme that usually the TTG should convert gliadin to glutamic acid. But since you have antibodies against TTG, so gliadin is not converted to glutamic acid. Okay, so celiac disease only occurs in people who are positive in, again, genetically predisposed. If you have HLA, DQ2, and HLA, DQ8. So you are genetically predisposed to have celiac disease. Okay, so what are the symptoms of celiac disease? Symptoms vary with age. So usually adults and older children or teens may also experience unclassic symptoms such as arthritis. So yung mga older adults can develop arthritis, neurologic symptoms, iron deficiency anemia, skin manifestations. Okay? So, but however, common symptoms would include diarrhea, abdominal pain, bloating, and others. So, how do we diagnose celiac disease? We should be able to detect IgA TTG. Detection of IgA antibodies to TTG. So this is the screening to determine whether you have celiac disease or not. So serologic testing for gliadin antibodies was first developed, but it is not used commonly today because it is not specific. So more recently, the test for deaminated gliadin peptides, okay? So the DGPs. So DGPs stands for deaminated gliadin peptides, okay, have been developed, and these DGPs, deaminated gliadin peptides, are actually sensitive in young children. Okay, sensitive in young children. So there are also tests that can determine the celiac disease, particularly if you develop for EMA has already been developed. So EMA stands for endomysial antibodies. And endomysial antibodies are important because it directed against the layer of connective tissues surrounding the intestinal muscles. So this one is very specific. This one is very specific for celiac disease, but are costly, okay? Because you need to do the indirect immunofluorescence. So biopsy of the small intestine should be performed to confirm the diagnosis. Okay? So that is for the celiac disease. Okay. So aside from the celiac disease, we also have the autoimmune liver diseases. Okay? So autoimmune liver diseases would include immune-mediated liver diseases that can lead to end-stage liver failure if untreated. So this one could actually be very fatal. So clinical features would include elevated liver enzymes. So what are examples of elevated liver enzymes? So this includes your SGPT, your alkaline phosphatase, okay, your lactate dehydrogenase, among the others. Okay, fatigue, nausea, vomiting, abdominal pain, itchy skin, and jaundice. So one common examples of autoimmune liver disease is known as the autoimmune hepatitis. So the autoimmune hepatitis is also known as the chronic, formerly known as the chronic active hepatitis. Okay, so there are two types of chronic active hepatitis. So we have the AIH1. So usually it's positive for SMA and ANAs. So 2/3 of the cases are actually AIH1. And then the AIH2 is positive for LKM1 or LC1 antibodies. And this one is usually seen in female children. By the way, SMA is the smooth muscle antibodies. So smooth muscle antibodies, I think we have already discussed this in anti-nuclear antibodies a while ago. Smooth muscle antibodies are directed against the myofilament known as actin. So another component of the cytoskeleton. So usually we can determine this using indirect immunofluorescent assay. And we are using the substrate coming from the rodents. Again, rodents ulit, no? Rodents, kidneys, stomach, or liver. Okay. And then there's another antibodies that you are not familiar with, and this is the LKM1. It is the liver-kidney microsomal antigen. So LKM1 stands for liver, kidney, microsomal antigens. Okay. And then we also have the primary biliary cirrhosis. So it's another example of autoimmune disease characterized by the destruction of intrahepatic bile ducts known as cholestasis. So majority of the patient this time will produce antimicrobial antibodies. So again, this is also detected by indirect immunofluorescent assay using the rodent liver, kidney, and stomach sections as okay. So that is the autoimmune disease. Now, another example of autoimmune disease is the multiple sclerosis. Okay, so this autoimmune disease, multiple sclerosis, is characterized by inflammation. So there is an inflammation of the and destruction of the central nervous system. Okay, so most patients produce antibodies against the myelin basic proteins, and plaques are formed in white matter of the brain and spinal cord, causing destruction of the myelin sheath of axons. Myelin sheaths are very important in our nerve because it will allow, myelin sheath will ensure that the impulse will travel smoothly, okay, in our to our CNS, to and from the CNS. So, but since there are plaques that are formed in the white matter of the brain and spinal cord, and there is a destruction of the myelin sheath of the axons, then impulses will not be able to travel smoothly. As a result, there would be visual disturbances, weakness in the limbs, dizziness, and sensory abnormalities. So, laboratory findings will include lesions as seen on MRI. And then there would be an increased immunoglobulin spinal fluid and increased IgG index. So what is the IgG index? IgG index is the ratio of CSF to that of the albumin to serum IgG. So ratio of CSF IgG to that of albumin to serum IgG and that of the albumin. So oligoclonal bands on protein electrophoresis of CSF is seen. So when you say oligoclonal bands, it means that two or more distinct bands are present during the electrophoresis. Okay, so that's what we meant by oligoclonal bands. So, as you can see here, multiple sclerosis is characterized by motor weakness, ataxia, impaired vision, urinary bladder dysfunction. There is a paresthesia, paralysis, mental aberrations, sclerotic plaques, and I think I have already explained the IgG, increased IgG in CSF, elevated levels of antibodies against measles virus, also in serum and CSF, and there is a suppressed T cells and an increase in B cells. So again, the myelin sheath of a healthy nerve doesn't have any sclerotic plaques or plaques. However, in multiple sclerosis, the myelin sheath, which is a single cell whose membrane wraps around the axon, is destroyed with inflammation and scarring. So there is what you call sclerotic plaques. Okay. So here's an illustration depicting the destruction of the myelin sheath by T cells. And since the myelin sheath is destroyed, so impulses will be impaired. So myelin sheath is intact, the message moves quickly, or the impulse is conducted smoothly. But in cases of damages, then the impulse or the message will move slowly. So that's why there's visual disturbances. You cannot walk normally. So that's the implication of multiple sclerosis. Okay, so there is already a therapeutic intervention for there is a therapeutic intervention, and that is by let's say the clizumab. So this particular drug, this particular monoclonal antibodies, and natalizumab will somehow prevent increased destruction of the nerve or of the myelin sheath. So here's an example of a MS patient because of the paralysis. So there's a tendency that the patient could be bedridden. Okay, so let's talk about myasthenia gravis. So myasthenia gravis is characterized by muscle weakness and fatigability because the depletion of acetylcholine receptor at the myoneural junction. So this one is characterized by enlarged thymus and antibodies against acetylcholine receptor. So that is the characteristic of myasthenia gravis. So symptoms include later on you'll be able to see drooping of eyelids, double vision, inability to retract corners of mouth, smooth muscle weakness, especially in the upper limb, difficulty in speaking and swallowing, inability to support head and trunk, and it can progress to affect respiratory muscles. So it's because it affects the neuromuscular junction, resulting in weak muscle, okay, weak skeletal muscle. So most patients have antibodies to AChR. AChR stands for acetylcholine receptors. Acetylcholine receptors. So it is a receptor on muscle cells that binds acetylcholine release from the nerve ending to cause muscle contraction. So acetylcholine is an important chemical to initiate skeletal muscle contraction. Okay, so however, because you have developed antibodies to acetylcholine, it will block binding of the acetylcholine, okay, to its receptor and transmission of nerve impulse that activate the muscles. So there won't be normal skeletal muscle contraction. So antibodies to AChR is traditionally detected by radioimmunoprecipitation. So there is a technique called radioimmunoprecipitation, which will detect antibodies against the AChR. So most recently by ELISA or by fluorescent immunopolarizing technique. Okay, so a small percentage of patients produce antibodies to mask. So some patients develop antibodies to mask, which stands for muscle specific kinase. So some patients would have developed antibodies to mask, and some patients would even develop LRP4. Another example of lipoprotein. So some patients develop thymoma. Some patients develop thymoma that contains autoreactive T cells. So here is another example of antibodies destroying the acetylcholine receptor. So acetylcholine, by the way, is a neurotransmitter that is important for skeletal muscle contraction. And so myasthenia gravis is characterized by orbicular oculi muscle that controls eyelid movement. So there's drooping of the muscle, and the masseter muscle is also affected. This one is being used to chew food. Yeah, so the drooping of the eye muscle, and the patients cannot really stand because of the muscle weakness. Okay, so let's talk about another example of autoimmune disease. This time, let's talk about the Glenbard Syndrome, which is characterized by the motor weakness first of the lower extremities, then at the upper extremities, respiratory muscle paralysis and paralysis. So there is a mononuclear cell infiltrate and demyelination. So just like your multiple sclerosis. So this time, you develop sensitivity of the lymphocytes to nerve extract. So somehow the patients develop anti-nerve antibodies. So just like your multiple sclerosis in Glenbard syndrome, there is a damage of the myelin sheath. Okay, so let's talk about Goodpasture syndrome. So Goodpasture syndrome, here the patient produces antibodies to basement membrane, particularly at the lining of the renal glomeruli and lung alveoli, specifically directed against the alpha-3 chain of the type 4 collagen. So in Goodpasture syndrome, immune complex will bind to basement membrane, and then it will attack and cause damage by type 2 hypersensitivity reaction. So if you remember the type 2 hypersensitivity reaction, this one is known as the cytotoxic. So immune complex will bind to the basement membrane. So again, so pag sinabi natin immune complex, it is made of antigen and antibodies. And then it will attract complement. And now it will cause damage of the cell. So it is known as the type 2 hypersensitivity reaction. So syndromes can progress to renal failure if the basement membrane is found at the glomerulus, or respiratory problem if the respiratory problem if the alveoli are actually affected. So if there's a respiratory problem, it is characterized by cough, shortness of breath, and hemoptysis or coughing out of blood. So antibodies to GBM is found in most patients. So we can detect it also by means of indirect immunofluorescent on frozen kidney sections or ELISA for antibody to alpha-3 subunit. So tissue anti-GBM detected by direct immunofluorescence on kidney biopsy sections will produce a smooth linear ribbon-like fluorescence along the GBM. So again, indirect immunofluorescence. So as in summary, Goodpasture syndrome is characterized by pulmonary hemorrhages, hemoptysis, and hemoptysis if the immune complex is found in the lungs. However, it can produce hematuria and acute glomerulonephritis if the immune complex is found in the glomerulus. Okay, so antibodies can either deposit in the alveoli of the lungs or glomerulus of the kidneys. So immunoglobulins appear to represent an antibody specific for an antigen shared by kidneys and lungs. So that's the reason why two organs are affected. So it is also known as the immune complex phenomenon. So this is the Goodpasture acute glomerulonephritis and the immunofluorescence imaging of linear distribution of an IgG on glomerular basement membrane. So this is an example of pulmonary hemorrhages if the lungs are affected in Goodpasture syndrome. So this is the reason why there is a, sorry, I forgot to move my slide, but then let me show again. This is the picture of a person with, or rather of the capillary of the person with immune complex. Okay, if these capillaries are found in the kidneys, acute glomerulonephritis could actually happen. And then this is the immunofluorescence image of linear distribution of IgG on glomerular basement membrane. And then this is the pulmonary hemorrhages in Goodpasture syndrome in the lungs. So if these are present in the patients with Goodpasture syndrome, then patients will be characterized by hemoptysis or coughing out of blood. And pernicious anemia is another example of autoimmune disease. It is the progressive destruction of the stomach glands. Okay, progressive destruction of the stomach glands. Gastric mucosa infiltrated with monocytes and segmenters. So here, you develop antibodies against the parietal cell antigen and antibodies against the intrinsic factors. Okay, so bullous disease is another example of autoimmune disease, but this time it affects the skin. Particularly, it is actually characterized by the deposition of immunoglobulin in squamous intracellular spaces. So, circulating anti-basement membrane antibodies are characterized by pemphigus vulgaris and bullous pemphigoid. So again, this is an example of pemphigus vulgaris. Here, you can see that it can happen in vaginal opening and also in the skin. And bullous pemphigoid is characterized by lesions in the skin. So it is an example of autoimmune disease wherein antibodies are directed against the squamous epithelium of the skin. And then polymyositis, dermatomyositis is another example of autoimmune disease characterized by weakness of striated muscles with muscle pain and tenderness of the muscle, characterized also by skin rash. So this one is characterized by hypergammaglobulinemia, deposition of IgG, and complementing the vessel walls of the skin and muscles. So yeah, this is how the tissues look like in cases of polymyositis, dermatomyositis. And another example is the acute disseminated encephalitis. So it is another example of autoimmune disease characterized by headache, backache, stiff neck, nausea, low-grade fever, weakness, and paralysis. So this time it is characterized by cell-mediated allergic response. So acute disseminated encephalitis is also characterized by accumulation of macrophages and other WBC at the gray and white matter. Experimental allergic encephalomyelitis can occur in the laboratory. It occurs 2 to 3 weeks post-sensitization upon exposure. Okay, so this one is characterized by perivascular inflammation. And Lte cells will cause demyelination or the destruction of the myelin sheath of the nerve cells. And of course, you do also have the hemolytic diseases. So as what I told you in the introduction, our own antibodies can destroy our own cells. So this type of hemolytic anemia, though it was already discussed in hematology, I just want to review it with you, is characterized by warm autoimmune hemolytic anemia if the temperature or if the antibodies becomes optimum at 37 degrees. But if the antibodies reaction becomes optimum at 4 degrees, then it is known as the cold autoimmune hemolytic anemia. And then we also have the PCH or the paroxysmal cold hemoglobinuria. This one is caused by DLA or the Donat-Landsteiner antibodies. The IgG is directed against the antigen. Then we also have the immune idiopathic thrombocytopenic purpura. Okay, this time the antibodies destroy our own platelets. Hence, it is characterized by PTK, bleeding problems, and platelets of less than 100,000. So remember, the normal range of platelet is between 150,000 to 400,000 cells per cubic millimeter, or 150 to 400 times 10 to the 9 per liter. So in idiopathic thrombocytopenic purpura, antibodies are produced against platelets, and this can be drug-induced as well. So there you have it, the different kind of autoimmune diseases. Thank you very much for listening.